Welcome to the first live virtual demo, again, for the BioProfile FLEX2 and the FAST CDV analyzers. My name is Trypan Blue, and I am the director for the bioprocessing portfolio at now Nova Biomedical. With me today doing the webinar will be Matt Miller, the PAT Bio North America Applications Manager. So let's get started. One thing I'd really like to make sure is that we would love to hear all of your questions during this demo. So please feel free to leave us a comment either using the chat functionality that should be on your top bar of your screen at any given point, and we will address them at various points during this demo. So feel free to engage with us with any questions that you may have during the demonstration. Before we get started, I just wanted to have a quick moment to introduce you to Nova Biomedical, if you're not familiar with us. So Nova Biomedical is a leader in the scientific diagnostic instrumentation, specifically on the bioprocessing and the clinical markets. On the bioprocessing biopharma side, we're a global leader when it comes to osmolality testing and whole blood biosensor development. One of the world's fastest growing IVD companies when it comes to our clinical market, serving our customers in patient care, and a long history in expertise in cell line development. Across the board, we at Nova Biomedical are focused on building technologies that provide insights to scientists and clinicians in making quick, fast decisions for what's best for their process as well as for their patient care. As some of you may or may not be aware of, Nova Biomedical was acquired by Advanced Instruments as of July of last year. And both of these companies have a rich history and decades of experience in their own spaces. So for instance, Advanced Instruments has its core foundation with development of freezing point depression technology for osmolality testing, cell line development with the Solventum portfolio, and liquid volume verification with RTEL. And Nova Biomedical have core foundations when it comes to biosensor technologies with clinical utilization in terms of stat profile and stat strip, and with the BioProfile portfolio focused on cell culture analysis and cell culture analytics in upstream bioprocessing. So together now as a combined business, we are known as Nova Biomedical, and we are focused in bringing best of both of these cultures and companies' legacies into this new identity that we have with you today. Now, today our focus is specifically on the BioProfile portfolio. As you can see, within the bioprocessing, we have 4 distinct portfolios, starting all the way from cell line development for single cell seeding, cell isolation, and clonality, proof of clonality with Solventum. We then follow through with the core BioProfile portfolio for cell culture analysis, where today you'll get a glimpse of the FLEX2, which is an industry leader and recognized for its value when it comes to cell culture analytics. And the newest addition or newer addition to the portfolio, the FAST CDV. And then you have the osmolality portfolio from Advanced Instruments Osmotech line that's focused on osmolality testing across the entire bioprocessing workflow. And lastly, RTEL MBS with the liquid volume verification for automated liquid handlers and pipettes. So with today's demo really focused on the BioProfile portfolio, want to give you a quick look at high level of what these two products are. So you have the BioProfile FLEX2, which is a comprehensive multi-parameter analyzer that can give you insights into 16 different key process parameters in under 4.5 minutes. You have the flexible option of having to do where you can introduce the sample manually on a carousel, online sampling, automated, or in a 96 well plate. And today we'll give you a glimpse into all of that flexibility that this device can offer. And you can get all of this insight with just a very small sample volume of 275 microliters. On the side, on the other end, you have the FAST CDV product. Quite like its name, it is fast, within 70 to 90 seconds, you are getting a full sense of what your cell density, viability, total cell count, aggregate looks like within your device, within your cell culture. It's a fully automated Trypan Blue based technology. And again, at the core, we do believe that small sample volumes, but giving you large insights is where our portfolio is focused on. All of these products are also designed in full understanding that instruments and decisions made upstream in process development will affect downstream and need to be able to be managed in both non-GMP and GMP environments, meeting all regulatory requirements, such as even 21 CFR Part 11 compliance. So that's a high-level view of these two products, but really getting into more and giving you a real personal look at these devices will be Matt Miller. Before I hand over to Matt, I do have a quick poll question to understand who's joining us today. From what part of the workflow are you in? So the poll question should appear on your displays now. If you could take a quick second to select your choice, we would be able to then capture that and make sure Matt is addressing it, targeting the mix of the audience that we have amongst us today. Right. I think that was sufficient time, given that was a very quick question. So thank you so much for taking that poll question. We are now going to switch to Matt Miller so he can get the demo going. Again, quick reminder, you have two options. Use the chat function, the Q&A function, leave us questions, and we will sprinkle those questions and answers throughout the demo, or if we don't have time, we'll always get to it at the end or reach to you even after that. So I'm going to stop here and hand over to Matt. So let me quickly turn the cameras around so you can see Matt. And you know what we're doing? We are keeping the instruments a bit of a surprise. So there we go. We do have the devices here for you. So I'm going to make another quick change to make sure you can hear Matt. Matt?
Yes. Can everyone hear me? Very well. Nice to meet everyone, at least virtually. Super excited to talk about the BioProfile line in this facet. Thank you, Shweta, for the introduction. I'm the National PAT Biotechnology Application Manager. I've been with Nova for about 8 years, and pretty much since I started here, I started working with the FLEX2 as kind of our core bioprocessing unit that we use for cell culture analysis. Happy to go into details of that instrument as well. Some of you on the call might also have some experience already with the FLEX2, and that's totally fine. Maybe there's some new questions or things that you have that we can touch on as well. Beyond that, the FAST CDV, as Shweta mentioned, is new to our product line, and so we'll go into a little detail on that aspect. Again, questions are needed anytime. We can stop and answer them. This is totally up to you in terms of the content as well. All right. Thank you so much. Let me go and talk about the FLEX2 first. All right. So the FLEX2 is our bioprocessing cell culture analyzer. This has full capability to do a bunch of different tests. So this is a lab in a box. You have a osmometer, and this is true freezing point depression osmolality, 48 samples you can load up at a time. You have the ability to do chemistry and gas on the system plus pH. You have a CDV, which that is our acronym at Nova. CDV is our acronym for cell density and viability using the Trypan Blue exclusion assay. And we'll go a little bit more on all these modules as well. Additionally, I have the OLS. This is the online sampler piece that is a relatively new piece to make an automated way of testing your bioreactors, your samples on the FLEX2 scheduled on the system as well. I think what I want to start is an overview is that this system is utilized for both all the way from cell line development to process development to pilot to GMP. And it's built that way with 21 CFR Part 11 compliance and all the integrity that we have with the software, the data, and the usage specs that we have on this system. A little bit more in terms of what this replaces. So if you have this system, you don't need the standalone pH meter. You don't need your BGA. You don't need your standalone cell counter. You don't need your nutrient metabolite analyzer. This is going to do all those and more all at once in the FLEX2 system, which is why it's such a such a good life in the industry so far. But we're happy to go into more detail with folks who are not as familiar with it. Initially, I want to start by just kind of talking about a little bit more of the specs here. So on the system, this is configurable in the field too. So if you don't have the osmolality piece, you don't have the cell density piece as well, that can be upgraded in the field quite easily by our service team. So that's a good piece to know for some folks. Looking at the screen here, the basic menus of the FLEX2, it's a touch screen, very easy to use with even double gloved hands. I can proof of that from going in some of these GMP sites that I visit. I can go back and forth with this with my finger. I have a keyboard as well as a digital keyboard as well. And then looking at the main screen, this is kind of your basis for understanding how the system is working at this moment. So you have the chemistry, the pH gas, the CDV modules, their status, how much life is left in the packs, how much sample is left in those sensor cards, as well as onboard quality controls, QC chemistry and QC pH gas. That gives the ability to test your sample before you run your precious samples. You can make sure that everything is working properly, and you can schedule this ahead of time, especially in a GMP setting where this is useful to schedule ahead of time, for different shifts and operators that might be using the instrument. Dialing a little more in terms of the specifics of those 16 parameters that Shweta mentioned earlier. We have glutamine, glutamate, glucose, lactate, ammonium, and a bunch of different electrolytes, as well as pH, PO2, PCO2, the CDV, and the osmo. So all these are hot buttons with drop-down menus to see the status of the packs. I'll show you one right now. So for instance, chemistry, I touch it, I see everything is nice and happy. We don't see any uncalibrated sensors, things that we don't need in that regard. The pack life, when the pack was installed, and we're going to dial in a little bit more by opening the box here and showing you the packs and the actual consumables as well. So that's some of the hot buttons there. From now, let's open the instrument and get you guys kind of inside the system. So if I open both of these doors, you can see kind of the innards, and very complex machine. Don't have to know all of it, of course, but it's nice to know, right? It's kind of interesting for me as well. So we have the Osmo 48 osmometer right to the left here. And then we have the big part of this system is the reagent packs and the sensor cards. So for instance, we have a pH gas calibrator pack. I'm going to pull this out. This comes in a box, room temperature. There's no prep needed for this. It's just plug and play in the system. And the reason because of that is because it has an RFID tag built into the system that automatically says the lot number, the consumables, what type of pack it is, when does it expire to the system, and on the audit trail as well. So very easy to install. If you haven't done it, you just put it in, push it, and you're set. You close the door, the smart RFID tag, the reagent is recognized, and then you're able to go forward with that and use the system. That's one of the reagent packs. Let's talk about the sensor cards. So Shweta mentioned the biosensors that Nova is known for. That's our core R&D legacy. This is biosensors. That's how we're able to get these small volumes that we can test with for pH gas, chemistries, and for CDV too. But focusing on the sensor cards for the chemistry module, these are credit card size sensor cards. Very easy to use, very easy to install. You pop this out of the fridge, your cold storage. This is a credit card size sensor card. Just let it sit here, put a new one in, pop it shut, close the door. The system does any kind of priming, hydration that's needed, and then it's ready to go on the system until the use life expires for this sensor card. So very easy in that regard. Just kind of a little behind the scenes in terms of what we're seeing here. This is a robot. So this has a lot of capability that we have a XYZ axis probe arm and a probe that senses fluid. And that's another reason we were able to get small volumes, because we have this probe that's able to determine small parts of volumes of fluid accurately dispensed throughout the system, including the chemistry, the pH gas, the CDV modules, and the osmo module as well. So that's a little bit inside the hood. We'll close this back up. Anytime you close the door, put in new packs, the system automatically does a whole maintenance thing and says, did you put something new in? Do we need to do a priming sequence? In general, the system is a very minimal maintenance system beyond just adding the packs, because you have the ability to have scheduled maintenance ahead of time to clean the system, to flush it, to make it as minimal maintenance as possible for you as the end user. Okay, let's talk a little bit about the interface and what you would normally work with as a customer. So regardless of where you are, cell line development, process development, you would start maybe with the analysis tab here. So this is a hot button. I can use my finger, I can use the keyboard. Let's go to the analysis tab. We have up to 4 modes of sampling depending on the FLEX2 characterization that you utilize. Initially, you have the syringe or manual sampling. We have a syringe there because ideally you're probably pulling a bioreactor sample from your bioreactor with a syringe and running that over to get a quick sample, the most accurate sample for pH, gas, and cell counts as well. You have the ability to configure and customize your sample types. And this is useful because maybe some other person in your lab has a different profile, different tests that they're running. You can create your own sample types and for dropdown and utilize what's needed. And the modules will be set up appropriately for that particular sample type. So if I go back to, let's say, default, that has all the modules, chemistry, pH, gas, CDV, and the osmolality module. One thing I haven't talked about quite yet is that we have the ability to do onboard dilutions. We have gigantic analytical ranges for both chemistry and CDV. And the way we're able to accomplish that is because this robot, this probe moving around the system, is able to properly dilute your samples on board. So you don't have to do any pipetting, any use of the consumables for pipetting to do those dilutions and get a very large range. So to give you an example what that range is for the CDV, for the it goes from, on the FLEX2, all the way from 100,000 cells per milliliter to 160 million cells per milliliter, all done automatically on the system with one shot with just a button selection. So that's really cool. That's another reason why I'm super excited about this system, and I have been for a while since I started at Nova. If we go to the carousel, so this is another way of sampling it. You can use these cups here that Nova provides. And when you select any of these cups, this is called the load and go carousel. So you can load some cups, you can have them sampling, and then you prepare more samples as needed, load onto the system as needed as well. You can see you can input sample ID, batch ID, anything that's useful to you, but you don't have to do that. This is all up to you and what results that you need for this system. That's the load and go carousel. If I switch to this button right here, the last one can be either a 96-well plate formation, and this system is built with the online sampler. So it takes the place of the configuration of the 96-well plate and gives you the online sampler piece. And we'll touch about that in just 1 second. But from a standpoint on the software, you have the ability to have 10 bioreactors sampling on one FLEX2. And that's pretty unique, I think, for the industry as well, that this FLEX2 can be working quite hard for this automated sampling. And I think at this point, I'm going to move to the online sampler, and we'll show that. Excellent. So the online sampler piece is to the right of the FLEX2. This box right here is the sample transfer module. Really, this is just the hub of where all the tubings come into the system. All the samples come into the system and get analyzed automatically. The way that's done is because we have, for each of those 10 bioreactors, or less if you don't need it, is an RSM, a reactor sampling module. This is a pump and all the fluids needed to clean that pump and push the actual sample to the system, the FLEX2, for analysis. This sits right next to the bioreactor. As you can see here, we have a 6-liter glass bioreactor. The way this works is you have what's called a reactor line assembly, and this is this little line here that's connected to the bioreactor. This reactor line assembly comes from Nova as such, and you can autoclave this up to 5 times or just throw it away after every use. It's up to you. That can be cleaned in between by you if you want to do that as well. This is an easy setup. and it maintains the sterile barrier between the bioreactor and the RSM. And the way it does that is because when you're setting this up, you're setting up your bioreactor, you're doing that, you're autoclaving it, you're setting this connection up maybe in a laminar flow hood. So you have this sterile connection to your bioreactor in your dip tube, right? The only piece that needs to be assembled in your lab space where you have your bioreactors running is this one piece here with an aseptic connection. And after every sample, you have a sterile air run through these pieces. That's why you see a vent filter on this side as well, so that you have a protective air in between your bioreactor sample and the FLEX2 as well. After every reactor sampling run, you have a cleaning, a flushing, and then stored in sterile air. So you have no ingress from your bioreactor into the reactor sampling module. So that maintains that sterile barrier, as I mentioned. We can configure to not just the glass bioreactors. We can configure to other bioreactors. It can be, we're very configurable, and that's where my application team comes in. So we can do configuration in the field of the CDV module. We can configure the online sample configurations for the different bioreactor types, because you don't only have just a 6-liter bioreactor. You might have a disposable bioreactor. You might have a bag that has a weldable fitting. You might have a larger bioreactor in the pilot scale. This is going to be really useful for you to configure and do automated sampling on any of those devices with our free, no-cost support from applications team. And that's something that is very unique to the industry, and that we're very proud of, that our applications team is out there assisting alongside our field service team, which is out throughout the nation and the world as well, assisting and helping your units. Okay, so that's a little bit of the online sample. Any questions at the moment that I can answer or help with? Yep. Sure. Consumables, yeah. So in general, the FLEX2 consumables, you need at least a chemistry and a gas pack and a chemistry sensor card and a gas sensor card for the system, but it's configurable. If you don't need if you're not doing cell density, you don't need a CDV pack. You don't need this Trypan Blue bottle pack here. If you don't have the osmolality piece, you don't need those osmo tubes either. But that's the basic feature of using the system. It's a wet analyzer, so you do need packs on the system for usage. There is the ability to shut down the system if you're not using it for a period of time very easily, and we can walk you through that process separately if needed. Absolutely. I think you're referring to, it's a good question. I think you're referring to the when I mentioned the sterile vent barrier. So I'll pull out this tubing that's assembled on here. This is the reactor line assembly. On both sides, there's a sterile, mLX filter. If a 0.25 micron filter that comes with this device, and then you do the autoclave process. Only at the point of putting this together on the system once for your whole process run, do you have to take off this one little filter and then assemble it onto the reactor sample module. So you do a little IPA spray right here, and then you have your whole configuration set up without any need for any playing around with any other connections. Hope that answers your question.
All right, we have one more question. How is a cell-free and cell-containing sample realized? I assume cell-containing needs to be used for CDV and cell-free for metabolite.
Not necessarily. If you have cell-free, you can run any of the modules. If you have cell-containing, you can also run any of the modules. So the sensor cards, you can get osmo readings with cell-containing samples as well, and you can get chemistry, pH, and gas samples or readings with cell-containing or cell-free. So I think that if that answers your question, otherwise I can dial in more on that.
Thank you, Matt.
Yeah.
So we have another question. Does the FLEX2 come already with a cell library, or do I have to feed the system accordingly?
That is a great question. So
we have built-in cell inspection types for the system, but this is where our no-cost application team comes in. because we want to customize the system for your cell parameters, what we call cell inspection type, CIT. We can also then look at if you have any other secondary third-party cell counters that are offline, we can correlate and work with the parameters we have to better match that if needed. That way you can use both as needed as well. And that's the idea behind that. When we come on site, we run your cells, we create a report and a summary to give you that information, help you implement that in your PD space and even GMP as well.
Thank you, Matt. A question there is PO2 and PCO2 still displayed with millimeter of mercury unit, or does it convert with the value into percentage?
Great question. That's a great question. So in terms of, let's go back to here, I'll show you that screen. So if I go to kind of the main menu aspect here, I can go to the settings and I can go to parameters and I can change any default from like millimoles per grams per liter, but you mentioned PCO2. So let's do that. So millimeters of mercury, kPa are the two settings that we have available to you. It will change your historical results automatically on the system for all the results that have been implemented in the past if you wanted to convert to just a different measurements.
All right, and one more question.
Absolutely.
In development, the expiry date is not always essential. Would it be possible to use a pack even if it is expired?
Yeah, that's a good question. So I think that's something that we can work with you on from a tech support or an application side. But once the pack is expired, you can't use that pack. But we can also look into and see what is your flow your flow through on the system. Are there times when you need to shut the system down and not use a pack? If that's part of your question, is like in terms of consumables and when I need to change and when not. But happy to answer more of that if needed or take that offline as well. It's a great question. Yeah.
All right, doesn't look like we have any more questions. So before we now jump over to the FAST CDV, we have another quick poll question for you. So you should see a quick poll appear on your screen. So if you could take a quick moment. This is to understand what may be a big challenge for you currently when it comes to cell density viability or media development workflows in your lab. We'll give it a few seconds here. I know it's a little bit more to read than the last poll, so. All right. Thank you. And now we're going to hand over to Matt Miller again to talk about the FAST CDV.
Excellent. Yeah. So this is our newest product to our portfolio, the FAST CDV. And as the name says, it's fast and it's the cell density and viability. So it is a similar Trypan Blue exclusion method that's used on the FLEX2. And it is 100 microliters of sample, so very low volume. You have the ability to do a carousel testing as well as a plate on the same system without changing these compared to some other competitors where you have to actually change them. You can use both at the same time if you want to. It's up to you. In terms of, you have the 100 microliter sample, the analysis time, as Shweta mentioned earlier, is 70 to 90 seconds per sample, so very fast. Fast, low volume. If you really care about those cell density results, in that regard, this is the unit for you. Now, one of the questions you might have is like, how does this compare to other instruments? And this was designed on purpose to compare to the FLEX2, but that's where the applications team comes in, no cost, with a system that you purchase, a system that's under a service contract, whatever it may be. We can do that legwork, that testing work to get better comparison as needed to all the different types of flavors of analyzers and cell counters that you have. All right. Similar to the FLEX2 on purpose, again, we wanted to make this easy to use for folks that are used to the FLEX2. We have a similar architecture here. So in terms of the buttons, it might look very similar. You have some of the same menus that you have on the FLEX2. I'm going to go into the analysis mode, which the screen that we're actually already in. now you can see we have the carousel and the 96-well plate. One thing I want to mention with the carousel, this is again, very fast carousel. Part of that is due to our tubing. So, sorry, our cups. So our cups are specifically made with plastic that prevents the adhesion of cells to the sides of the cups. And they're designed in a way that if you load up a full carousel, you can get proper mixing of that cup on the actual sample times. Because remember, just like the FLEX2, all the dilutions are done at the point of sample. So no need to do any pre-dilutions. Load up your sample, give it a go. The system will do all that testing. And the analytical ranges for CDV, for FAST CDV, is very similar to the FLEX2. You can go from 50,000 cells per milliliter to all the way to 140 million with just changing, I select this, just changing the dilution factor on the system here. So it's simple button change, or set that as a preset in your sample types. The same sample types exist, similar to what I showed you on the FLEX2. So that is one way of sampling, is through the carousel. We can also go the 96-well plate. So the 96-well plate, you have the ability to use your 96-well plate or our fast plate. And we call it a fast plate for the same reason that for the cups, is that we have that same plastic, that same design to have the better resuspension of cells, especially when you really load up that 96-well plate for accurate results. Let's take a look at this plate. Let's take a look at the configuration there. So if I open the drawer here, we already have a plate in here. So the only two reagents you need to add to the system is the Trypan Blue bottle packs, and I'll show you in a second the actual system pack, which is really just stores the waste. So here is the fast plate that I mentioned earlier. Load that up, bring that back, set that up on the system, close the door, and then you're ready to sample once you have your profile entered on the system as well. So that's really cool. You can do both of those at the same time as needed as well. Looking at the inside scope here, you're not going to see exactly the same stuff, but there's still a fluid sensing probe for that accuracy, and that high-speed camera that is able to determine the cell counts very quickly. That high-speed camera also uses AI and machine learning, which is part of that new update that Shweta mentioned for the FAST CDV that we're really excited about at Nova. The one pack that we have in here is similar to the pack that I mentioned before. Plug and play, easy to wipe down, sanitize, especially for GMP environments. Plug and play with that RFID tag, set it in the system, close it. The system will do the maintenance if it's a new pack, and go from there. I'm going to pause for just a second. There might be a couple questions that came in. If not, I'd be happy to move on.
Yep. No, we don't have any questions yet. So I think we're good.
Yep. So let's show you a couple results here. So we've got the idea of how to sample, and you can see that the system software architecture is very similar to the FLEX2. Similarly, you have automated maintenance on here, so it's a very minimal maintenance system in that regard. If we go to historical results, we can pull up your sheet of results. OPC is built into both this system and the FLEX2. So any transfer of results or other diagnostics can be done through OPC, can be done through Ethernet. You can have transfer of data from historical results to your LIMS system, your historian. Both of these instruments have a bridge computer built in, a host computer, and a CDV graphics computer. So 3 computers in one, and this is what makes this so fast and special too. If I look at one of these results here, I can view selected. We'll see this in a larger scale that may be a little bit more visible. We can see all the images. If we were curious and we wanted to dial in, which we're curious, let's do that. So let's take a look at this image here. We see live cells, the Trypan Blue exclusion assay. So the dead cells are going to be darker. The system learning is picking that up. We can also toggle live and dead off. We can zoom in if we want to. You can see your cells in a little bit better clarity here as part of your checks. Okay. And I'm actually going to show, for the last piece, I'm actually going to show the cells on the FLEX2 as well. Apologies with the camera changes here to move around to give you the best look here. Similarly, historical results in the FLEX2, you have a lot of parameters mentioned, and that same connectivity for exporting your results. You also have the ability to pull up your results here, and then to see either export or print out from here, or take a look at your results on this screen too. So these are similar but different way of giving you the live and dead indicators for the FLEX2, right? On a large screen, so you can easily view it, compared to some of the other screens might be a little smaller and harder to understand. Okay. I feel like that's a pretty good overview of the FLEX2 and FAST CDV. I'm happy to dial into something more if Shweta or other folks need some different details to be discussed.
Thank you, Matt. I really do want to open the floor up to all of the attendees. If you have any other questions and would like us to take a closer look at anything else, please let us know. Matt, that's been very helpful to get a glimpse between the two, the FAST CDV and the FLEX2.
Yeah.
And would this OLS module that you showed on the side be the same module that would enable the FLEX to connect with the ambr from the Sartorius?
Yeah, very similar. So beyond the OLS, which is connecting to your bioreactors,
I have a strong partnership with Sartorius for the ambr connection. So if you have an ambr 15, an ambr 250, a modular benchtop ambr, we can connect quite easily in a similar type vein where we have instead of an RSM, we have another pump on the side called an ESM, external sampling module, that'll then grab the sample and push it to the system here. Small volumes, gas impermeable tubing, accurate results. We can test those accuracy results. If you need that from our applications team, we can test that from both different sampling methods as needed to help you verify and qualify these instruments. Yeah.
great question.
Thank you, Matt. We do have one question from one of the attendees that was specifically focused on the FAST CDV.
Yeah.
They wanted to know if we had, we already had bridging studies that showed FAST CDV versus the Trypan Blue.
Yeah.
We do have some bridging studies that show that data. Happy to provide that for you. I think what probably would make the most sense, if you're really interested too, is that we can come on site and do that analysis. For evaluations, we can bring both of these instruments on site. We can run your cells. We can do that bridging study. We can make those cell inspection parameter correlations so you get the best feel of how this works for you, your process, your cells.
Thank you, Matt. And that's the case with the other viability analyzers in the market as well. We do have data, and we're happy to, as Matt has said, be on site and really work with your lab, your needs, and customize as needed. All right. Want to make sure we have any more questions as well from the folks on the call. Great. Okay. Well, at this point, we just want to make sure that this quick note, thank you, Matt, really very much for giving us a glimpse at both of these devices and what they can do for customers in the cell culture workflow. A note for all of the attendees, this will be recorded and available on demand on our website. So please feel free, for those of your colleagues that weren't able to attend, to take a quick look at this when it's recorded and available online. Additionally, you can always reach out to us with any questions using our contact form online, or reach out to your local key account managers, who will be happy to assist you with any of the questions, as well as consider having somebody on site for an evaluation, demo, or even comparison bridging studies, as we've discussed previously. But this has been a wonderful experience. Thank you so much, Matt, and thank you all for taking the time to join us today.